the time to present their work. And Dr. Berry comes from us from MGH, and I know that some of you knew on the program that Dr. Sokovitz would be here, but he has kindly taken her place. She had a busy schedule and couldn't make it today, but I'm confident that Dr. Berry, who works closely with Merit, is involved in many of the trials at Niels. We talked a little bit this morning about the Clinical Trials Network with Niels, and we're very pleased that he's here today to give us an overview. So thank you very much, Dr. Berry.
Give sort of an overview of the principles of trial design and then talk about some of the clinical trials that are going on in ALS currently. So there are a couple of ways that we come upon candidate drugs to treat ALS, and really for any disease. The first is what we call targeted development. What we do is we look at a potential disease mechanism and we look for a drug that can interrupt that disease mechanism and restore normality. And the second is clinical observation. So it may be that a physician or a patient notices that a patient does better on a certain drug, and we can then test that drug for repurposing for disease modification. Both of these are viable strategies. Both of them are used. An example of the way that clinical observations may guide development is Idec. This was a drug that astute patients and clinicians noticed was helpful for the treatment of pseudobulbar affect. And it's now FDA approved after going through trials. We have many candidate drugs at this point, and really our goal now is to design trials and move through these candidate drugs and find the ones that are helpful in the disease. So novel drug development, if only it were as simple as discovering a new molecule and putting it into people, there are a number of steps that we go through. First, we do the basic science. So discovering a new drug structure and then figuring out the mechanisms that we need to produce this on a mass scale can take years of research and be very costly. And that's before we ever get to testing. This is all on sort of the very basic level. But once we do have a good drug structure, one that we think is theoretically going to be helpful, we can move into animal studies where we're looking primarily at safety data. But also some dose ranging to understand what dosages we might try in people. And we may get a hint about efficacy in some of our models. This may be a shorter process than the initial development, but it could also take a great deal of time. We then go to the FDA for an investigational new drug approval. And this is what allows us to move that drug into people. And the first thing that we do in people is called a phase 1 trial. And this is really a small group of people. We give the drug at a number of dosages, and we see if it's safe as sort of a first pass. These are known as first-in-human trials. They may be done in patients. They may actually be done in healthy volunteers as well. After this, we move to phase 2 trials, where we try to gather more information about the safety and tolerability of the drug. And we also try to get information about what we call dose ranging. That is, what's the best dosage to move forward to a larger trial and really look at whether the drug works for the disease or not. Usually these trials are bigger. 25 to 250 patients is a good estimate. And they're a little bit longer, following patients for months up to a year. And then the last stage really is to do a phase 3 trial, and this is to look at the efficacy, which is just another word to say, does the drug have effectiveness to fight the disease? And after the phase 3 trials, we can go back to the FDA, and we'll review this for a final approval, and then we can put this into people. And there is a way to speed up this process by repurposing drugs. In other words, taking a drug that's FDA approved for a different indication and moving it into the ALS space. So the basic science has been done. Many times, much of the animal safety studies has been done. And we can go immediately to the FDA for an investigational new drug approval. We can then move into people, and again, a lot of times the phase 1 data has already been gathered. This has already been put in people for the first time. And what we need to know is it safe and tolerable in a population of patients with ALS? And what's the right dose to move forward in our disease? We can then move on to the phase 3 categories. And so we really are putting our emphasis on the efficacy trials rather than doing all that background work. And then we would go to the FDA and they would review for approval. So this is a way to really speed development if we have available compounds that we think we can repurpose. Just a word about basic trial design. Sort of the paradigm that we work from is the same for any trial. We have a lot of permutations of this, but basically we take patients, Through a process called randomization, we randomly assign them to receive placebo or study drug, and then we measure clinical outcomes. So I want to talk about placebo for just a moment. Placebo is an inactive substance that looks exactly like an active study drug in a study. And part of the reason that we use placebo is because of the placebo effect. That is, our brains are wired to look for cause and effect in the world. And so if we take or do something that we expect to help us, we may perceive a benefit from that. And so that's one of the reasons that we use placebo. But there's another compelling reason to use placebo, and that is that a placebo group is a valid comparison group. So let's imagine for a moment that we did a study, and we gave a group of patients a new drug, and then we followed their ALS functional rating scale over time. And we found that this was the slope of that ALS functional rating scale. If we don't have a placebo comparison group, we might say, we don't know if that was effective or not. We can't be sure. People tell us they feel better, but we're not 100% sure. We might say, let's go back to the patients who are in the clinic who chose not to be in the trial. And we'll compare how our patients in the trial did and how those in the clinic did. And perhaps we would come up with a slope that looked like this, and we would say, based on this data, we would say those slopes look about the same. This drug was not effective. If we had a placebo group, what we might find is that actually people who were on placebo really did significantly worse. And this leads us to a different conclusion, which is that the drug is helpful. Now, why would that happen? It may be that people who were enrolled in the study had a more rapidly progressive disease course than those who did not enroll in the study. So in many ways, having a placebo group protects us from declaring drugs ineffective when they actually are effective. And that's, I think, a very important point. That the placebo information in a trial is as important as the study drug information in determining whether a drug is safe and effective. Why randomize? Well, the goal of randomization is to be certain that the patients who receive placebo are the same on every demographic and baseline characteristic as the patients who receive active study drug. And we want to ensure that the only difference between these two groups is the active study drug. And this is so important that when you look at published clinical trials, table 1 in every case is a table of demographics that demonstrate to the reader that the two groups were balanced, that the same kind of patient was in the placebo group as was in the active study group. Because without that, all of the conclusions from the trial can be questioned. So I'm going to talk briefly about the sort of phase 1, 2, and 3 trial designs. Again, there are permutations on that initial structure that I showed you. But in phase 1 trial, this may be the most different. We work in what are called cohorts. So we might have a first cohort that has as few as 3 patients. And we start at the lowest study drug dosage that we're going to test. And oftentimes this is one where we saw no adverse events in animals at all. There was no effect of having the drug, and there was no adverse effect of having the drug. And then we follow safety. And this is really on a very basic level. Is this bad for people? We then move to a middle dosage or a higher dosage, and we might move to a third dosage. And we keep going up to our target dose or until we see an adverse event. After this, we can move to our phase 2 trial. And in phase 2 trials, we involve more patients. We do use randomization here, typically to placebo or one of multiple study drug dosages. And the outcome that we follow primarily is safety and tolerability. And this may be followed by asking people about adverse events, may be followed by looking at laboratory studies. But we're also selecting a dosage through this study for our phase 3 trial, which is the one that will say yes or no, this drug helps for the disease. So we're going to select either the high dose, the medium dose, or the low dose to go forward. And the last thing is, sort of just like a child and a few days before their birthday, we may take a peek at efficacy. We can't resist. So we do include efficacy outcomes. We want to know, is the drug working? But typically in phase 2 trials, we don't have enough people to prove that beyond a doubt. So regardless of what we see, we often have to go on to a phase 3 trial. And likewise, even if we don't see efficacy, even if we're not sure that the study drug is making a big difference, we still often want to go on to a phase 3 trial because we just don't have enough patients to prove one way or another based on a phase 2 trial about whether it's effective for the disease. phase 3 trial is where we involve many, many patients. It's a much simpler design. We randomize the study drug to placebo, and then we follow our efficacy outcomes. Now we also follow safety, that's an important part of any trial. But our real goal here is to find out whether this drug works for the disease. So based on that, I'm going to talk about some of the ongoing trials for ALS. And what I'm going to do is try to categorize these by disease mechanism that the trials are targeted to. So I've drawn a motor neuron, and you'll see on the right-hand side of the slide the arm from a second motor neuron. And those two are not connected, but they communicate through a neurotransmitter called glutamate. So it's a chemical that bridges the gap between the two motor neurons. And at the bottom of the slide, I've drawn a muscle just to orient you to the slide. So the first disease mechanism is called glutamate excitotoxicity. Cells must have a neurotransmitter called glutamate to communicate with one another, but if there's too much glutamate, it can be toxic to motor neurons. Ceftriaxone is a drug that's being tested now. It's a repurposed IV antibiotic. It's aimed at reducing the amount of glutamate and thus excitotoxicity. And this is being tested in a continuous design for phases 1 through 3. So rather than doing phase 1 and stopping and analyzing, phase 2, stopping and analyzing, and then phase 3, it's being wrapped up all into one to try to speed the development. Phases 1 and 2 did show safety and helped with dose selection for the phase 3 trial, and the phase 3 part of the trial is ongoing now. Enrollment is complete, and that trial is aimed to end at the end of 2012. The next disease mechanism is called protein aggregation, that is, clumping of proteins in the cell, and for example, we see this with SOD1. One trial that's aiming to alleviate this problem is the ISIS 333611 trial. This is a phase 1 trial that recently completed. This is a gene therapy to turn off the SOD1 gene or turn down the SOD1 gene. And it's being tested in patients with familial ALS due to mutations in the SOD1 gene. Arimoclomol is also aimed at reducing protein aggregation of SOD1. This is in phase 2/3 trial. And it increases the gene expression of chaperone proteins that we hope will clear the protein from these clumps. Again, it's being tested in patients with familial ALS due to SOD1 mutations, and this trial is enrolling currently. One of the problems that is ubiquitous in ALS is muscle wasting and atrophy. And there's a drug, maybe not targeted exactly at this, but really more at the weakness of the disease, called CK357. Which has just completed phase 2 trials. A number of phase 2 trials were done, and I'll talk in a little more detail about it in just a moment. But it's an oral drug that's aimed at improving muscle contraction, the efficiency with which a muscle contracts. This is a symptomatic treatment, but I think we'd all agree that it's targeting a very important symptom of the disease. The next mechanism is inflammation-mediated damage. So there are, we think, inflammatory cells that are speeding the progression of the disease. We have one trial that's going on now that's targeting this as a disease mechanism. This is the NP001 trial. It's a phase 2 trial. This is an IV infusion drug. It's given for 3 days each month over 6 months, and then people are followed over a period of 12 months. The drug is aimed at reducing overactivation of immune cells. There are many kinds of immune cells. In this case, it's monocytes that are being targeted. Enrollment is complete. This is an ongoing study, and it ends in late 2012. Maybe missing the bottom of no. So fingolimod, we're planning a phase 2 trial. This is an oral drug. It's approved in MS, but it does have known side effects. It's aimed at reducing circulating lymphocytes, which are a different kind of immune cell. But again, it's working on the inflammation pathway of the disease. RNA dysregulation is becoming a very important topic in ALS. And just to take one step back, DNA is sort of the blueprint for the cell. DNA makes RNA, and RNA makes protein, which is the building block for the cell. And so if that process becomes dysregulated, we don't have the right proteins, we don't have the right building blocks for the cell, and it can cause illness. Right now, we don't have any trials that are targeting this, but my guess is that we're going to have many, many trials in the future. It's a very complicated process, and we've just begun to learn in recent years about its role in the disease. Mitochondrial dysfunction has a tremendous amount of important work supporting its role in the disease. We have a couple of studies that are targeting this. The first is creatine tamoxifen. This is a phase 2 trial. Both of these are oral drugs. Both are aimed at improving cellular energy metabolism. Tamoxifen is also aimed at changing hormonal response on a cellular level, I should say. This is what's called a selection design trial. In other words, there is no placebo, and this is one of the rare kinds of trials where we don't have a placebo. The two drugs are being run against each other, and whichever looks better then would be taken forward to a phase 3 trial. Dexpramipexole is in phase 3 trial. You'll hear a little bit more about this one in just a moment. This is an oral drug aimed at improving energy efficiency in the cell. It's going on globally. There are about 900 participants, and the enrollment is complete. The study is ongoing and will end in late 2012. And the last sort of thing that I'll hit on is oxidative damage, and we don't currently have any studies that are fighting that specifically, but we do have a couple of studies that are aiming to support the cell as it undergoes whatever stress it may undergo. One is Neuralstem, which you'll hear more about soon as well. This is a phase 1 trial in which stem cells are transplanted into the spinal cord to try to support the motor neurons that are there. And this is enrolling by invitation currently. Brainstorm is a company in Israel that's running a phase 1 trial of bone marrow precursor cells, which are taken from a patient and then manipulated to secrete what we call growth factors, which are supportive of motor neurons, and then transplanted into muscle and CSF. And again, this is enrolling in Israel. A couple of words about CK357. This is a drug that's being supported by a company called Cytokinetics. It's being tested in patients with ALS. It's just completed phase 2 trials, in which 3 active doses were compared to placebo. In part A, patients were off riluzole, and part B, patients were on a half dose of riluzole, so 1 day dosing of riluzole. There is an interaction of the 2 medications, and so that was part of the reason for the designs of these studies. These studies showed that the drug was well tolerated both with and without riluzole. Dizziness was the most common side effect, but this tended to be transient, and that there was some trend toward a signal in the ALS-FRS as well as one of the pulmonary functions. But again, remember, these are very small trials and we wouldn't be able to prove one way or another. It's enough evidence to say that a phase 3 trial is warranted. ISIS 333611 is a gene therapy to reduce SOD1. It's what we call antisense technology. Again, DNA makes RNA, RNA makes protein, and in this case, protein is the problem because it's a misfolded or mutant protein. The drug binds to and reduces the RNA for SOD1, thus decreasing the protein. It's specific to SOD1, so it's only active on that protein. And this trial, this phase 1 trial, was conducted in patients with SOD1 mutations. The drug doesn't cross the blood-brain barrier, so it had to be given into the spinal fluid. Just to give an overview of the study, patients were screened and then they were admitted to the hospital for a 12-hour infusion, not IV infusion, but actually into the spinal fluid through a lumbar puncture. Drug levels and safety labs were checked both then and 8 days later. There was an interim phone call, and the last follow-up was 29 days later. This was a fairly typical phase 1 design, except that the cohorts were bigger than usual. There were 8 patients per cohort, and there was a placebo in each cohort as well. There are a few, I think, worth mentioning non-drug intervention trials. One is the exercise study. So right now we don't know exactly how to make recommendations about exercise, and we're guided off of our experience and what we've learned from patients over the years. But this study is really aimed at comparing usual daily exercise to a regimented program, and will help us really guide our recommendations in the future. That one is enrolling now. There's a planned study of the diaphragm pacer system. This has what's called an FDA humanitarian use exemption. That is, there's not enough evidence on the FDA, in the FDA's view, to say that it's helpful for the disease, but there's not enough evidence to say it's harmful either. And so under the ethics board, for each institution, it can potentially be used currently, but we'd like to have an answer about whether it's helpful or not. And so there is a planned trial for that. There's also a nutrition study that's going on now. This is enrolling currently. It's being done in patients who have a gastrostomy tube, and it's comparing usual diet to one that's high in fat and calories. And the results of this may be generalizable to a much broader population. So clinical trials allow us to translate the amazing discoveries that you've heard about today, the that are happening in the lab and in the genetics realm into advancements in clinical therapy. And it's really your participation, the patients, the caregivers, that allows us to move forward. So I want to thank you. I want to also talk about a couple of resources that are available to help you understand what's going on. So one thing is that the Northeast ALS Consortium has teamed up with ALS's Treat ALS initiative with the goal of translating basic science into ALS research and clinical trials. And NEALS was founded in 1995 with 9 centers. It's grown to 100 centers. It's funded in some part by the Treat ALS initiative. And some of what this initiative does is allow young researchers to enter the field, allows resource sharing between ALS centers for clinical trials, ensures reliable trial results, which means we can depend on the results that we get out of our trials. It attracts biotech to the field by saying, we have a mechanism for testing drugs, therefore it's to your benefit to come into ALS and bring your compounds into ALS and test them here. And our hope is to provide patients with information about becoming involved in research. And to that end, NEALS has a new trial website. This is the NEALS and Treat ALS website. It's searchable. The website is alsconsortium.org. You can look for trials. It's a very slick search engine. And the other resource that I want to point you to is the trial expert who's available to answer your questions. You can email or call directly, and the toll-free number is listed, as is the email.
Thank you so much, Dr. Berry. I know that set the stage for the next 2 trials that we'll hear about, and then we'll open up for questions. So we're pleased to have our next speaker, who I'm not seeing, but I know he's close by, Dr. Carl Joe, who will speak to us about the Neuralstem trial.
Thank you very much for inviting me. I'd like to talk to you about the clinical trial that's going on at Emory University. Let's see if I can I supposed to point this at something? Oh. This one? Oh, yeah, I see the pointer. But how do I advance it? So much for the scientist, huh? This is, as you see, the very first slide shows you the title of the study that's ongoing. It's a phase 1 study, as Dr. Berry just described. It's an open label, which means the patients as well as the doctors know who's receiving the treatment, who's not. In this case, it's a single-arm study, which means all the subjects in the study are receiving the treatment. It's the stem cells, or specific, I'll describe to you in a minute what kind of stem cell it is, but it's a first exposure in humans. So we had to go through with the FDA a series of discussions about the safety aspects of this treatment. And the purpose is to look at the feasibility and safety of this particular treatment. And I would say I'm very proud to say that this is a model. I feel like it's a model collaboration between academicians and industry. I had a number of researchers, especially Dr. Feldman from the University of Michigan, Dr. Jonathan Glass from Emory University, and Dr. Bolus, also from Emory University, who conduct many of the preclinical studies with us together that led to the initiation of this study. So I would like to describe to you where the cells come from, because there are many stem cells, so-called stem cells, in the media, and I would like to just very clearly point out where our cells come from. Our cells come from developing nervous system. What that means is that we harvest the cells from fetal tissues, which were obtained from elective abortions. The mothers donate the tissue to us for research and for development as drugs. And this particular tissue, or this particular cell line that's being developed, is derived from a fetal tissue that was donated to us back in the year 2000. We isolated neural stem cells from that one piece of tissue. We were able to expand it over many, many generations and be able to bank the cells, which means generate large amount of cells, freeze them down, characterize them, so that should the cell prove to be beneficial, we'll have millions of doses to be able to treat patients all over the world for the next many, many years. So that's quite different from mesenchymal stem cell or MSC cells, where the bone marrow derived cells are harvested from the patient's body and then filtered or manipulated and then put back into the patient's body, which is called autologous. So that's the key difference between allogeneic, which is what our cells do, and autologous, which is what a lot of stem cell clinics provide. Also, what makes a key difference is that allogeneic cells, because it's derived from single genetic background, going into many different genetic background, it requires immune suppression. Whereas autologous, from a patient derived going back into patient, does not require immune suppression. So in our trial, we have a rather serious immune suppression regimen that's mandated to the patients. We are diligently looking for data to support for ways to cutting back those doses. But I'm hopeful that we have those data coming up soon. So the origin of the cells illustrated here in this picture here, which is a picture of a mouse, fetal mouse, is this darkened area in the central nervous system. And this particular protein or gene, which is called anestin, is expressed in all of the central nervous system, from eye to the cerebrum, to the hindbrain, to the spinal cord, all the way down to the end. So you can imagine that this is where we derive our cells, but in human equivalent. And another thing that I would point out then, in essence, what we're doing then is to take this fetal primordial precursors of the developing nervous system, which will give rise to the mature brain and the spinal cord. We expand them in culture, and then we put them back into adult environment, that is specifically a diseased adult environment. The expansion of the cells allows us to characterize the cells very carefully. And this gives us a tremendous control over what type of cells we get, how to test them, and how to demonstrate their purity, potency, and in our manufacturing process. I won't go over the technical details, but in the end, that allowed us to generate a clinical lot of cells, which is in jargon is called GMP cells. And these are super clean scrub cells that allow the FDA to allow us to put into human patients. And as I said before, we were able to generate many generations of these cells, but not too many, but sufficient enough to be able to ensure many years of supply of the cells should it work. We have also put the cells into several different animal models of diseases, one of which was ALS. And this work was done in collaboration with Dr. Kolyatsos' lab at Johns Hopkins University. And as you are well aware, the SOD1 rats have ALS-like symptoms, and injection of the human neural stem cells into the spinal cord of the SOD1 rats were able to prolong the lifespan of the rats, and as well as delay the onset of the rats. The primary mechanism of how the cells achieve this benefit appear to be